Okay. So thank you very much, Bernard, for the introduction, and thank you for the privilege of speaking here in this wonderful setting, and it was a great morning. So, as Bernard said, in my laboratory we are interested in understanding the importance of cell-to-cell communication in bacteria and how they regulate behaviors in multi-species communities. So, as we've already started to hear this morning, biologists are very aware at the moment of the importance of the microbes that live inside and on the surface of our bodies, and there's a huge amount of work, especially studying the microbes in our intestines or in the mammalian intestines. And however, despite the large amount of a body of literature that supports the beneficial properties of our microbiome that go way beyond just nutrition but also behavior, I think what we are at the stage where most of the studies are still very based on correlations, and there's a lot of question marks, especially in regards to understanding the molecular mechanisms that are behind these beneficial properties that our gut microbiome can bring to our the health of its host, either being human or other mammalians. So in my laboratory, we are especially interested in understanding the beneficial property of colonization resistance that microbes have as a protective barrier against invasion of other microbes like pathogens. The natural microbiota confers a natural protection against the invasion of these microbes, and it's in this type of properties that I think bacteria-to-bacteria interactions are more important, and that's that's where we are making more. We are addressing our studies is really to look at these interactions here. Oops. So this is a cartoon made by one of my students, Anahita, which I like very much because it illustrates very well the way we in our lab imagine the mammalian gut. At the moment, as I said, we know very little about the molecular basis, and basically at the moment we know who's there. We know the different species that are present, and these molecules here, these chemical molecules here, represent molecules that we know that these microbes are producing. As you can see, there's a lot of question marks. But for me, the way I see this is the most interesting multi-species ecosystem that we know of, because there's hundreds of different bacterial species that need to coexist and interact with each other and with the host. And we like to understand how this process is occurring at the molecular level. So these molecules are quorum sensing molecules, which we learned from in vitro studies that in vitro they've been shown to be involved in regulating what we call a quorum sensing process that regulates bacterial group behaviors. Very briefly, the way this works is that at low cell density, like this stage here, these molecules are not detected by the bacteria because they diffuse away. But at high cell density, the concentration of these molecules is high, and they're detected by the bacteria, triggering a response at the level of gene expression, allowing bacteria to synchronize their gene expression like in a multicellular tissue and engage in group behaviors. So these molecules that are these quorum sensing signal molecules basically allow bacteria to regulate gene expression as a function of the density of the population, and we call them autoinducers because they are produced by the bacterial cells. So we say that autoinducers mediate cell-to-cell signaling in bacteria, allowing them to regulate group behaviors. And these group behaviors include a variety of things. They were first discovered in this symbiotic marine bacteria that produce luminescence, which is an example of a type of quorum sensing process where it's only productive to produce light when they're at high cell density. In specific case, they live in symbiosis with this squid. Here we have Pseudomonas aeruginosa, a human pathogen that synchronizes the production of virulence factors by sensing the presence of these quorum sensing signal molecules, and we have a plant pathogen that regulates the production of extracellular enzymes to degrade the tissue of its host. In this case, it's a potato, and it does so only when it's at high cell density, when it can sense the presence of this molecule. And we're learning that bacteria can sense this molecule not only to regulate in terms of density, but also because these molecules have different species specificity, they can also determine if they are within a polyspecies community or in a monospecies community. So they can use these molecules both to know how many cells are around them and what type of species composition the community has. So from the work that we and many others have done in vitro, as I said, we've learned that these molecules are extremely important to regulate these bacterial behaviors, which are only productive at a high cell density. And given the huge numbers of bacteria that we have in our intestines, and the fact that some of these bacteria we know are producing these molecules, we've asked how is this playing a role in the colonization of the mammalian gut. And what I'm going to be telling today, what I'm going to summarize to you, is the work we've been done to establish a model where we can manipulate the levels of this signal molecule here called autoinducer 2 and start asking the question of the importance of this type of cell signaling in bacteria in the mammalian gut. So why autoinducer 2? Because it's this molecule here is the only molecule that we know it's produced by a wide variety of bacterial species across very distant phylogenetic groups. This autoinducer 2 molecule is produced both by gram-positives like Bacillus, Lactococcus, Streptococcus, but also many gram-negative bacteria like Escherichia coli. And we've shown in vitro that it can foster interspecies communication by regulating gene expression. So you can mix different species and they can regulate each other's gene expressions through this molecule, so it can foster interspecies communication. So this is why we thought would be the perfect candidate to study in terms of the context of the mammalian gut. And because it's from in vitro studies, we know it regulates many different quorum sensing-like behaviors, like production of biofilms. There's this interesting case of phage dispersal in Enterococcus faecalis, or virulence in Vibrio cholera, motility in E. coli and Salmonella, as just a few examples. So, as I said, we are interested in asking what the importance of this molecule in the gut microbiome, and in particular in the beneficial properties of the gut microbiota in colonization resistance. So this is just a very simple experiment to illustrate to you what we're talking about in terms of colonization resistance. So when you have a healthy microbiome, it's very difficult to colonize with an invader. And this is what Nahita, my student, is doing here. She has an E. coli strain. Here in blue she colonized orally a group of mice with a high dose of Escherichia coli, E. coli, and then she measured the colony forming units on plates by plating the fecal samples from these mice. And as you can see, after day 1, E. coli is basically washed away and could not colonize the mouse. But if you treat the mice with streptomycin, in this case, an antibiotic in the drinking water, E. coli, and we keep the streptomycin, E. coli will stray at very high levels, because this is also a streptomycin-resistant strain. And what is very important here is that when we give streptomycin, we're not just simply killing the microbes. We are actually changing the composition of the microbiota. And so you change the composition from a resistant microbiota community to a susceptible microbiota community. And this is what I'm showing you here. This is the phylogenetic composition of the gut microbiota at this stage here before starting the antibiotic treatment, and this is upon streptomycin treatment. So before treatment, we have what we typically have in a mammalian gut, which is this very important proportion between bacteria that belong to this group called the Firmicutes, which includes several Clostridium and other gram-positive bacteria. And you have here this very specialized group that appears in all, as far as I know, all mammalian guts, these bacteroidetes. And this one-to-one proportion between the Firmicutes and the bacteroidetes is important for many properties of the gut microbiota, such as colonization resistance, but also nutrition imbalances and other problems that can occur under situations like this one, where you have what we call a dysbiotic state, because now here, after the streptomycin treatment, we have a huge expansion of the bacteroidetes. I'm not showing you here, but we also have the total levels of these bacteria, and what happens is that antibiotic is not just killing these bacteria. Now you have the same number of total bacterial load, but you just have a much bigger proportion of these bacteroidetes. And now these mice are very susceptible to E. coli, as I was showing, but also to other pathogens. So what my students, the postdoc Jessica Thompson and my student Anahita did in collaboration with Carl Zobeda, who helped us starting with the microbiota analysis, was to ask if we manipulate this autoinducer 2 quorum sensing molecule in the gut, will this affect the antibiotic-induced dysbiosis in the mammalian gut of mice? So in order to determine if autoinducer 2 would have an influence during this process, they had first to establish a way to manipulate the signal. And to do that, they took advantage of a system we had discovered some time ago in E. coli that has a very particular system that allows it to produce this signal, but also to degrade the signal. This is here in black. What I have is just in vitro cultures of E. coli. This is a wild type strain that doing growth will first produce the signal and then degrades the signal. And I've studied this process in great detail, and we know how it's controlled. It's controlled by this transporter system, and we can make mutants and engineer this E. coli strain to accumulate different levels of autoinducer 2. And for the purpose of today's talk, what matters is that we can have a mutant here, this blue mutant, it's a kinase, that now does not degrade the signal. So it accumulates to high levels of the signal. And we have this red mutant, it's just in the repressor of this operon, that is very efficient in removing the signal from the environment and degrading the signal. So we can call this, we use this mutant as a mutant to scavenge this autoinducer 2 and this other mutant to produce high levels of the signal. So, and we had shown that in vitro, you can manipulate quorum sensing of other species using these E. coli strains. So they were engineered and optimized to colonize well the gut of these mice, and they put these strains in the mice and determine how this would affect the autoinducer 2 levels in the mouse gut. Specifically, they were looking at cecum extracts, and we weren't sure what was going to happen with the wild type because it can both produce and degrade, but it also accumulates high levels. These are the basal activity, and we have this blue mutant, the mutant in this kinase that does not degrade the signal, accumulates high levels as we were hoping, and then this mutant that degrades the signal really removes, we find no activity when we have mice colonized with this mutant here that is very efficient in degrading the signal. So then they had mice that were treated with streptomycin to induce this dysbiosis situation, and then they colonized the mice in 3 groups with 3 different E. coli strains. So the first one is the one I mentioned with this mutant that produces high levels of the signal. The other group is the scavenging strain that removes the signal. Then we have a control group that's also colonized with E. coli strain. And in this case, is a strain that neither produces, neither degrades the signal. So that's why we call it the control strain. So then we, after 1 month of streptomycin treatment, we collected DNA from the fecal samples and determined the microbiota composition in the 3 different groups of mice. And I'm just really just giving you the punchline, but what happens when we looked at the microbiota composition, here I'm showing you a principal component analysis where each dot represents the microbiota diversity in one mouse. And what we have is that the dots in red are from the mice that were colonized with the scavenging strain. The dots in black are the control strain, so they're completely overlapping, but we have a different microbiota composition in the mice that were colonized with this E. coli that increases the signal levels. So what this principal component analysis tells us is that the microbiota composition of the mice that had the E. coli that's increasing the signal had a different microbiota composition than the other mice. So it also tells us that we don't see difference, and we looked at different particular species. We didn't find a single one where it was different, increase the control strain or removing the signal. So there was, in this particular model, we didn't see a difference for removing the signal, but we saw differences by increasing the signal. And then, of course, we looked at this carefully in terms of species composition. And then the most interesting results were when we looked at the highest phylogenetic level. When we looked at the phylum level, we saw that the mice that were colonized with the E. coli strain that produces high level of these signals, they had more Firmicutes than the other 2 groups. So again, removing the signal didn't have an effect because it was the same as the control, but the ones with high levels of AI2 had more Firmicutes and slightly less bacterioids in the group of that had high levels of autoinduced 2. So this is telling us that AI2 is favoring the expansion of the Firmicutes and hindering the expansion of the bacterioids. So to make this really simple in a cartoon manner, what we had was, in the beginning, before treatment, we have more or less the same proportion of bacterioids and Firmicutes. What the streptomycin does is that it really kills most of the Firmicutes, but having the E. coli producing the autoinduced 2 favored the persistence of the Firmicutes. We still don't know the mechanism, but what we're seeing is that having the E. coli producing this quorum sensing signal really benefited this group that had been more affected by the antibiotics. So it is counteracting, we can say that it's counteracting the effect of the antibiotic. So this is so the other thing that was so this showed that indeed increasing the levels of autoinduced 2 could affect the composition of the microbiota, and it was interesting for us that it was counteracting the effect of the antibiotics, and but we are looking at these changes in microbiota composition during antibiotic treatment, and in the follow-up that Anahita is looking is really to what comes out of this experiment is that it suggests that AI2, this autoinduced 2 signal, might help accelerate the recovery from the antibiotic-induced dysbiosis. So what she's doing now is looking at stopping antibiotics, allowing the recovery of the microbiota in the presence or absence of autoinduced 2, and we hope we'll see a faster recovery in the presence of the signal, because this is what this experiment already indicates. So the other thing that was very interesting to us is that so we could identify that autoinduced 2 is favoring the Firmicutes, and when we looked at the genomes of these bacteria, we saw that the Firmicutes are the ones that have 80% of this LuxS gene. It's the gene that is responsible for producing the autoinduced 2 signal. So Firmicutes have 80% of them are producing the signal, but the bacteroidetes, most of them do not produce the signals because only 20% have the synthase gene to produce these signals. So the other thing we've learned is that AI2 is favoring the producers. And this also allows us to start really identifying which are the bacteria in the gut that are responding to it. And the way we're moving forward with this is to simplify the community, to ask, we want to know, so the Firmicutes are being favored, but what are they regulating? And we are using not germ-free mice with the streptomycin treated flora. Now we can put this in germ-free and then colonize E. coli. Now we don't need the presence of the antibiotic, and we allow expansion. And what we saw is that there are some groups like this Bacterial, for example, that's at very high levels in the population. It's at 10% of the community, and it's responding very well to the presence to manipulation of the signal and this Suturella. And so now we can do experiments with to identify really which are the direct interactions, and by looking at gene expression data, we can look at the behaviors that are being regulated. So just to summarize, we have established the methodology to manipulate the interspecies signal in the mouse gut. This leads to the hypothesis that AutoInducer2 can accelerate recovery from antibiotic-induced dysbiosis, which we are looking at, and we're doing Gnotto mice and RNA-seq to look at to determine which are the direct interactions, which species are interacting directly, and are they going to be regulating biofilms? Are they going to be regulating secretion of hydrolytic enzymes, as they do in other, like in plant pathogens, for example, in order to understand which are the functions that are essential for gut homeostasis and which are important in this beneficial microbiota community. Okay, so this is just my group, and Rita, and Jessica were the ones who did most of the work I talked to you today. Thank you.
Thank you. We have one question from Jean-François Bach.
Well, these are very interesting data. Maybe I would like to ask a further question. We have used antibiotic-treated mice now. There has been a number of studies using probiotics, including E. coli, in models of autoimmune diseases and allergic diseases. And the results are not always straightforward, but by and large, there is a protection from the disease using these probiotic preparation. Notably, there was a paper in Cell last year showing that E. coli derived from the gut of patients with high incidence of diabetes had a lower capacity to protect NOD from diabetes than E. coli from patients who were resistant to diabetes. So my question is, in non-antibiotic treated animals, just let's say normal mice without treatment, what is the effect of such probiotics, particularly E. coli? Do they change the gut microbiota composition? Do they have other effects? What would you say? Because it just does not apply only to E. coli, it applies to other probiotics.
So I don't know if I understand correctly, but you are surprised that in some cases it's making a beneficial effect, and in some cases it's detrimental. But so you have to remember that E. coli it's extremely versatile organism, and there are many strains of E. coli, and we all have commensal E. coli, in our intestines, but the diversity of E. coli is tremendous, and some are really very pathogenic, right? But so that's one point, right? I don't know in those two studies that you're referring to if we're talking about different strains of E. coli. And it's so some produce toxins, some don't produce toxins, right? And some are some can become intracellular pathogens, and some don't have. They can vary up to one fifth of their genome can be different. So they can have very different effects in terms of what they do. I think it's also very important to remember that E. coli is usually present at very low abundances in the gut, but it's very versatile again, and so if upon disturbance it can reach very high levels. So it might be beneficial at very low levels, but it can be detrimental at high levels. So you need to look at the different strains and the abundance that they achieve in the gut. I don't know if that helps.
Daniel Louvar is on the back here, right here.
Very nice. You showed us the effect on the E. coli population and the microbiota. Did you ever investigate in your experimental design whether or not you have a change of the turnover of the epithelial cell of the gut and on the different proportion or the different ratio or the different cell type of the gut, particularly with respect to goblet cells, for instance? No. So
we've been really just focusing more on the microbiota composition, but so for me this study was really nice to be able to show that yes, the community is responding, but now it also allows us to understand who's responding more, and it allows us to identify how to have bigger responses. So now we're focusing in trying to have even bigger responses, because then it will be a challenge for me to start looking at the questions you're asking, but I wanted to have a system that had a greater impact than what we're seeing now, and then we really will go into all the outputs of the host that we can remember and where we have expertise and help to do it. Right?
Thank you.
Philippe Sansonetti on this side. Eric.
Sorry. Yeah. So the kind of dysbiosis that you obtained is kind of obtained by a very drastic approach, which is treating with a bactericidal antibiotic. So would you dare to expand what you said on other situations in which the dysbiosis might be a little less drastic? I would say, like, you have animals that have chronic inflammatory status and they have some level of dysbiosis. You can create situations by changing the diet. Are you thinking about milder situations and generalize the AI2 hypothesis?
Yeah, so we are thinking in looking at other dysbiotic situations in models for Crohn's diseases, for example, where we don't have such a huge change. But even in the antibiotic, yes, we did learn that this was we did this because we started with a I didn't want to manipulate the microbiota too much to make it very homogeneous, but then what happened was that antibiotics really can disturb the community in very unpredictable way, and so the noise was tremendous, right? So we saw a signal only when we had a very long treatment, because the response was more homogeneous between the different mice. There was less mouse-to-mouse variation. But what we're doing now is that we understand the system a bit better. We're doing co-housing to normalize the initial state, and we realize we need to use shorter doses of antibiotics, because otherwise we were not seeing a recovery, and so we'll continue to play with shorter doses of antibiotics. But yes, you're right. Lower dysbiosis is probably better. Yes.
Eric Westhoff, down
yes,
there.
Thank you. Is it known to which molecule this auto-inducer 2 binds? Because Firmicutes, they contain a lot of riboswitches, etc., so that regulate gene expression?
Yes, so that's also one of my favorite. I'm a microbiologist, and we really started by looking at the receptors and signal transduction pathways. So in the synthase to produce this signal molecule is highly conserved among these different species, but the receptors are not so conserved. But we, Bonnie Basler had already discovered the receptor in the vibrio. Then together with Steve Miller, who's also in this picture, we identified the receptors in E. coli and Salmonella. They're present in gram-negative bacteria. And we saw a few examples in Bacillus where there's evidence for lateral gene transfer. So we know the receptor in some of the Bacillus. And now that there's more genome sequence, we started to look at the receptors again, and especially after this data, we're very interested in the Firmicutes, and we identified the first receptor in Clostridium, which was fascinating because the binding site is not 100% conserved to the E. coli one. There's some 2 amino acids modifications, but we have the receptor, so now we can do more molecular targeted. But yes, that's a very important next step to understand the molecular mechanisms that the bacteria are using.
A short question by Pascal Cossard and a short answer.
Okay. So can you use the synthetic molecules and increasing the doses and just see what is the effect?
Yeah, so the molecules is a bit unstable. We're working with the organic chemists to do a prodrug. They call it prodrug because it's protected by a sugar that only then degrades in the intestines, and yeah, we're doing that, but it's not there yet.